All right, let's get started. I'm Bill Louv, president of Project DataSphere, and I'm your moderator for today's webinar. Project DataSphere has two primary activities. One, we operate a data sharing platform, a rich source of clinical trial patient-level data across the spectrum of cancers. If you go to our website, you'll find it's very straightforward to get access to a very rich body of data. We also operate a portfolio of data science-driven programs where we bring together experts from multiple disciplines, from academia, from industry, from government agencies, to solve problems and improve outcomes for cancer patients. One of our research programs is in external control arms, and that's the topic for today. We brought together 4 experts who are part of our effort in that research program. And in turn, each of them will introduce themselves and make some comments about this research program. What are the opportunities? What are some of the challenges of this area of using external control data? We're also going to dig deeper into one particular application area, which is glioblastoma, and look at some innovative clinical trial designs as well as the use of external data. So we'll go ahead now to our experts. We'll all then come back together in a panel discussion. Just a reminder, this is recorded, so we can use it for at other times. Also, we love your participation through the chat box at the bottom of your screen. Enter any comments or questions you have in there, and we'll touch on those when we get to the panel discussion. So our first expert, our first speaker is Dr. Jacob Sands. Jacob, over to you.
Great, thank you so much. I'm Jacob Sands. I'm a thoracic medical oncologist at Dana-Farber and chair of the External Control Arm Task Force. Thank you to those of you that are attending this, and of course to those watching the recording at a later time. So I'm going to provide just kind of a brief overview as to this, but we'll get into some of the more specifics as we go further down the line. Essentially, science is happening faster and faster, and so innovations are therefore happening faster and faster. And what we're seeing is that there are studies being done where at the time that they're developed, the control arm that makes sense at that time is no longer the control, is no longer considered the standard at the time of publication. And we've certainly seen this within non-small cell lung cancer recently with publications comparing to a control arm that is outdated and no longer utilized. And this really raises the importance of more rapidly accruing trials and getting to these answers more quickly. On top of that, of course, we're all working toward reducing costs of trials and being able to bring innovations forward faster and faster. And so what we're going to discuss in this is an external control arm, which is a way of utilizing previously accrued data, whether that be in trials or collected data, even from real-world setting, but previously collected data with the details that allow for true matched controls, and then being able to compare our experimental arm in a way that provides a bit more context. That's more detailed than if we were to utilize historic controls, which of course have their own challenges. On top of this really push for how rapidly we want to move things forward and the need for that, there are rare populations where trying to actively accrue a trial that truly includes a control arm and an experimental arm is quite challenging due to the rarity of the diagnosis itself. And so this has really brought forward the need for this concept of an external control arm to then be able to utilize data that is detailed enough to really be matched controls for an experimental arm going forward. Now, as we go forward, I'm going to turn it over to Frank in a moment to provide a bit more of a framework as far as regulatory. And then, of course, we're going to get into some of the specific details as to the building of this itself. And with that, I will turn it over to Frank.
Thank you, Jacob. Frank Rockhold. I'm a professor of biostatistics and bioinformatics at Duke University Medical Center. I sort of came into the picture due to my interest in data sharing, which is how I got engaged with Project DataSphere. But I also have an intense interest in study design. So this project has been a great interest to me. What's interesting is that if you look at the history of external control arm data, Stuart Pocock published a paper in 1976 suggesting the concept of using external control arm data in cancer trials. And of course, back in 1976, nobody thought of an electronic health record yet, or what we call real world data, but the concept still applied. And so there was some basic concepts in there that says a lot of what we're doing today is sort of an extension of that. We obviously have a lot more and better data than we did back when Stuart was doing that work. And we have two kinds of data. I'm going to simplify and say we have real world, what we call real world data, and I'm defining real world data as health application data, so claims or electronic health record. And each of those data sets have versus clinical trials, and each of those data sets have different provenance and history and where they came from. And I'll give some specific examples. Now, one of the points I think that the regulators are focused on for good reason is their substantive evidence guidance, and I'm thinking particularly the FDA here, says that they have a particular interest in different study designs. And if you look at their rules, I mean, randomization rules as guidelines, I should say, randomization plays a key role in that. And so how do you then marry that? And that's certainly was true even the updated 2019 guidance, and even in the 21st Century Cures Act said that substantive evidence guidance still has to be upheld. So how do you create study designs? And as Jacob said, and particularly in diseases where it's hard to find concurrent controls. So one of the things this group has done is tried to characterize sort of a range of study designs. So if you sort of were thinking about a sliding scale, a classic randomized controlled clinical trial would say be of minimal risk, but entirely contained within a clinical trial. All the way at the other end of the highest risk scale is a single arm trial with no control. So all there's options in between. There's an external control arm using clinical trial data, such as from Project DataSphere, which is probably the middle of the road. External control arm from real world data, which would be considered higher risk from real world data. And then a design that I'm particularly interested in, the hybrid design, where hybrid design is a way to marry the concept of an external control arm data with a sort of a randomized trial. So the principle is I'm just, and as an example, you only randomize half of the control arm patients, and you extract the rest of the information from a clinical trial data, external clinical trial, real world data source. And then you use the randomized portion to sort of update and create a better matching scheme. Of course, randomization by itself is inherently creates matching. It also, the randomization, I think one of the reasons it's appealing to the regulators is that it also deals with unknown confounders and also keeps the investigator honest in terms of putting patients into the trial. And for adverse events, the use of randomized control data is actually easier in terms of matching for adverse events. So there's this hybrid design, I think, has a lot of appeal. You don't have to use 50%, you could use 20%, and that's, I think, Lorenzo has got a lot more expertise. He's going to present some technical details of different designs. But the concept is you can retain what looks like a classic randomized trial, but reduces the amount of weight you put on the people actually being concurrently randomized, makes it easier to meet some of the needs that Jacob mentioned, but at the same time, utilizing external control arm sources. So I think it's a great way to utilize existing randomized control data. And one of the reasons glioblastoma is a good use of those data is that the treatment hasn't really changed much in the last few years. So even if the trial is a few years old, it may result in the ability to use a reasonable control. And it also helps meet what I think is a great need to be able to utilize data for data sharing, which I think will then promote the concept of data sharing. People can see a good use from it. So I'm going to stop there, and I'm not sure who's the next one speaker, Jacob? Is it Lorenzo?
To Lorenzo to discuss some of the specifics.
I just want to make sure. Okay, thanks.
Thank you, everyone. And I'm Lorenzo Tripa. I work at Dana-Farber, where I started working in glioblastoma together with a PI of a large adaptive randomized clinical trials in glioblastoma. And then since then worked with neuro-oncologists at the Farber. And then thanks to Project DataSphere, I put together this group of scholars, we continued working on this idea of integrating external information into clinical trials. And so there are a couple of ideas that I'll try to summarize and express with support of a couple of figures. Here is what I consider the key decision problem when we design randomized or non-randomized trials. We perfectly know that in terms of resources, level of evidence, we have on one extreme single-arm design. The main weakness of single-arm design is the rigidity. Does not match inclusion into final analysis about the context, the type of population that has been enrolled, whether it includes an older population than another trial, whether the performance status of the patients tend to be high or low. And on the opposite extreme, you have randomization, which eliminate, and it's considered the gold standard to face these problems of possible confounding. And here in the middle, I put a single arm design in which we try to do the best use of external control. And the key problem is to understand, are we close to the operating characteristic of randomization, or are we more in the neighborhood of a single arm design, and basically with the same weaknesses and same problems of potential bias and risk of false positive and false negative results. So in the last few years, a couple of years, we tried to use collection of data sets in order to answer this question. And obviously the answer is, it really depends on the data that you have. Low quality data make a single arm design with an external control extremely weak. And we need to have tools, statistical tools, in order to realize whether we can have operating characteristic, low level of bias, which we hope to be comparable to a randomized clinical trial, which require much larger amount of resources and patients. Here, another figure. The main point I want to make here is it is not true, as Frank was mentioning, that we can use external data only to replace randomization. There are several other great things that we can do into trial to make them more efficient. Here, there's one example in the bottom of this slide, which represent a hybrid design. That's what Frank was mentioning. On the top, another very simple idea. Those yellow bars are just interim look at the data. So you have a large randomized clinical trial. We should use all information we have in order to make a good interim decision. And the decision could be to stop the trial if the drug is not promising, or other decision that can be made is to expand the sample size when we feel it's needed. As I was saying, we try as much as possible to build on previous experience, previous data sets. And for us, it was quite relevant, this meta-analysis that we published in neuro-oncology a few years ago. Here we got data summary on over 400 studies. And what we realized was a large proportion of patients in glioblastoma were allocated in single-arm studies. We know the weaknesses of those studies. We try to understand where do they come from, what are the key mechanisms that bring bias, and can we have an answer to the question, why do we have also so many randomized phase 3 studies that failed in glioblastoma? So this is the meta-analysis. We try to address this question I summarize the result of the paper just with one single figure. From the meta-analysis, we can quantify the level of study-to-study variability. So to which extent the populations enrolled in different trials, they are different. In glioblastoma, that level of heterogeneity is definitely relevant. And that translates into high type 1 error, high type 2 error when we look at the operating characteristic of single-arm trials. So to me, this is the key motivation to try to do externally controlled trials in glioblastoma. And in fact, we have a large number of patients, a good proportion of those enrolled into trial that go into single-arm studies, number one. And number two, we have a high level of type 1 and type 2 error of these studies. So that's the key motivation, to try to incorporate external information and try to improve those characteristics. Here is the main idea that we proposed in order to go into that direction. We need to validate, not only to propose statistical tools, but they have to be validated. And we published this in Clinical Cancer Research in 2019. And in 2021, Rif published a review of possible approaches, and this is published in the Lancet Oncology. The idea in this validation scheme seen to the right of this slide is the following. You take a completed randomized clinical trial. We know they require large resources, but we can use them as the gold standard. And then we isolate only the experimental arm and combine with an external data. Here, it is represented in green. It can be a real world data set. At that point, we can pretend to reanalyze this single arm study with an external control. And obviously, if we obtain a similar treatment effect estimate, similar inference on the treatment as with the gold standard, that means we are in the right direction. And of course, one validation, it's not enough. That can be just an anecdote, and we repeat the same type of comparison for different randomized clinical trials. Here, the next figure that I show you, this is just how the data look like. You have different columns. These are many studies. Some of these columns are randomized clinical trials. Some are real-world data sets. And this is the collection of data we use in order to do validation studies. The primary figure is this one. We take 5 randomized clinical trials, and we do that validation analysis that I discussed earlier. Out of those 5, we would have 2 false positives with the single arm trial. And this problem of bias and potential false positive get substantially reduced when we incorporate causal inference tool, here represented in blue, and the idea of incorporating external information. Here, the summary. First of all, we need meta-analysis, data collections in order to evaluate the clinical trials. So whether they incorporate external information or other type of innovation, it is always possible to start from the previous experience in order to have projections of what is the trade-off between bias, type 1 error, and power, and the resources that need to implement the trial. This has to be done, and unfortunately, it's context specific. That means we can have a positive answer, and the figure that I showed you from Clinical Cancer Research manuscript, that tell us going with external information in glioblastoma is a good idea. That doesn't imply, obviously, that the same strategy works in another disease. It can be an horrible disease in gastric cancer or in other diseases. But we need data collection that are specific of the disease, we need them to not become obsolete. And that's why we work with Project DataSphere. The other thing that I want to really emphasize, we started with really simple statistical designs. Just a single arm, and we add the external information. We use tools from causal inference, and we try to understand if that is a good idea. But the nice thing is that there is a galaxy of possible trial designs, hybrid design or part of it. And a nice thing from a statistician point of view is that they have been largely unexplored. I see opportunities to be creative and to create efficient clinical trials in the upcoming years, incorporating external information. And from this, I stop here. Thank you so much, everyone.
Okay, Rif.
Great, thank you. And thank you very much for having me today. I'd like to thank Project DataSphere and organizers for putting this on and letting me participate. I am a CNS radiation oncologist in Boston at Dana-Farber and Brigham and Women's Hospital. Clinically, I treat tumors of the central nervous system, and my clinical and research interest is in glioblastoma. So in thinking about and sort of setting the stage, I think the motivation for pursuing this idea of external control arms for GBM, for glioblastoma is feeling that it is an area of tremendous opportunity for this approach. As we all know, GBM is truly a terrible disease. So even with maximal treatment, meaning maximal surgery, chemotherapy, and radiation therapy, median survival is estimated to be approximately 15 months. So GBM is incurable, and recurrence is felt to be inevitable. So certainly an area of great need in terms of improved therapies. As I think Frank mentioned, for GBM, the standard of care has not changed substantially in many years. The current standard of care was really established back in 2005 with the STOOP trial, which was a randomized trial randomizing patients to radiation alone versus chemoradiation, and it showed a modest but real survival benefit with temozolomide chemotherapy back in 2005. Since then, there's really been very few treatment advances for GBM, and regulatory approvals are minimal. And I think the lack of progress is not for a lack of trying in the field of neuro-oncology. I think there continue to be tremendous efforts to develop better therapies. And I think what we've come to realize and acknowledge is that with all the number of possibly promising therapies that come out, we need to be able to evaluate them effectively and efficiently. Looking at sort of the drug development pipeline in GBM, as Lorenzo mentioned, there's been many failed phase 3 trials. And what we see looking back is that we're often using single arm trials into early phase settings, and that's led us astray and led us to these large, very resource intensive phase 3 trials that would show inevitably that the drug didn't work, given our lack of progress. So I think this has really been a big problem in neuro-oncology for many years now. And I think, the problem has been that we know that single arm trials are flawed, but we also know that randomization is very tricky in GBM, particularly in the early phase setting. Quite simply, patients don't want to enroll onto randomized trials and risk receiving a standard of care therapy that leads to suboptimal outcomes, and often it does not work. Especially for something that's felt to be incurable. So any kind of randomized designs often can lead to accrual and dropout issues, sort of the traditional randomized designs. So I do think that external control arms are a very appealing solution, specifically for GBM. And I really do think there's a strong rationale here and why it is growing interest for GBM, but in neuro-oncology in general. I think when we think of the reasons, again, as mentioned, we're historically have relied heavily on single arm trials, which we know are very limited. And so certainly a strong rationale there. We haven't made the progress we want. And as a byproduct of many failed trials is that we have many high quality data sets that are out there of patients treated with the standard of care for GBM specifically, since the standard of care hasn't changed. Furthermore, I think when we have newer treatments, we're thinking of more novel ways of approaching GBM, given that the standard ways don't work. And often blinding or randomization can be extra challenging for these novel therapies that are coming out and being tested in GBM. And then finally, I think, given how dismal the outcomes can be in this disease setting, I do think that trial designs that can minimize the number of patients randomized to the control arm of a trial in a way that is still not leading us astray from a statistics perspective, I think needs to be thoroughly explored. And I think is another reason why it's really of gaining interest in this field, because we know that, you know, we don't want patients to be going onto the control arm of trials if, you know, more than needed. So with all those factors, I do think there's a very clear rationale here in terms of using external control arms for GBM. I know Lorenzo spoke to some of the methodologic work that, you know, at least provides us that I think is very promising and shows us that it seems that this appears to work in GBM. You mentioned it's context specific. It may not work in a different cancer or a gastric cancer or whatnot, but, you know, in GBM, it does seem to be very promising. It is already being applied in the neuro-oncology field, given this interest. So there's a company, Medicenna, that has completed a phase 2 trial in recurrent glioblastoma that's used an external control arm. And they are actually amidst planning a registrational trial using the hybrid design that Lorenzo described, which allows for less patients being randomized to the internal control arm compared to a traditional randomized design. So certainly something that's very exciting. And to my knowledge, it's the first registrational trial that will be using and incorporating an external control arm as part of its design. And it's something that the company and that touts that they've discussed with the FDA, and that the FDA was open to this approach and did feel that this would be an acceptable way to evaluate their therapy. So I think all of this is pushing towards the fact that there's a lot of buzz in neuro-oncology. You know, we've certainly, you know, had publications, we've had very positive interactions, whether it be with the FDA or with relevant stakeholders in the field as well. I think at Dana-Farber, you know, in our work with Project DataSphere, we've been trying to be at the forefront of this in the neuro-oncology sphere. And we've been obtaining high quality data from several different data sources that Lorenzo alluded to. And this allows for that methodological work, which I think is so important. And we're really trying to position ourselves to be a resource moving forward, so that the hard work we put into obtaining data with Project DataSphere can be used by the neuro-oncology community as a whole moving forward. So I think from my perspective, you know, there's several things we need to do in our initiative, including continuing to obtain and free data so that, because it's such an integral part to using external controls, is having high quality data, continuing our work with Lorenzo and the great work they do in terms of examining the methodology and ensuring that we're validating our approaches and doing things in an appropriate manner. And then, of course, continuing to engage with relevant folks. And it's been great to see how engaged everyone really is in neuro-oncology and on a regulatory perspective. So I think there really is a lot of excitement in the use of external control arms in glioblastoma specifically. And certainly I'm excited for the rest of the discussion here. But certainly it's been great to see.
Thank you, Rif. I especially appreciate you, putting that kind of clinical context into why it's hard to do the trials in glioblastoma, right? So you got this informative censoring. So even if you can recruit people, the benefits of randomization are sort of eroded in these trials. That's really important to understand that. So I was wondering, and I think all of you would have a comment on this. So how is this looking with respect to our friends at the regulatory agencies? And so is the analysis plan for Medicenna, is that what we've come to expect for an analysis plan? So there's an interim evaluation of the randomized versus external control group, and then there's a branching there depending on the outcome, or is it still, yeah, you can do that, and we'll tell you what we think of it once we see it? So I just wondered, that's part A of my question. How specific is that analysis plan with Medicenna? And the second is, are there other examples of people bringing hybrid designs to the FDA? or other regulatory agencies? Maybe the scene is different in Europe.
Maybe one comment on this. Well, I participated in some discussion with the FDA and with the EMA, the European agency. First of all, the impression I had, which I can share, is it really depends on the disease. And it is a key factor to understand how difficult it is to have a randomized trial for a specific cancer type, for a specific population. And that really matter on the position of the regulator. The second factor to consider is that the discussion on this type of design incorporating especially real world information is relatively recent, which make legitimate and perfectly appropriate for the regulator to seek a gradual experience toward that direction. So this is sort of my personal prediction, but I think the class of designs that Frank was mentioning, hybrid, meaning incorporating some randomization and including the external information, it can be very attractive for regulatory purposes, because they are a way to combine some advantage of external information and randomization together, and it can be relevant in this gradual adoption, especially in the regulatory phase of drug development of external information. Mm-hmm. So I think it will be a transition, and it will be gradual. So there's a continuum of designs between just the poor randomization, and on the other extreme, a single arm just compared with an external data set, and we will see multiple points along that continuum.
Okay. Thanks, Lorenzo. Rif, I saw your hand up.
Oh, I don't think I that might have been by accident, but I can speak to it. So yeah, I agree. So, and certainly I think there is this openness, and it depends, as Lorenzo mentioned, very context specific. You know, there was, it seemed, I believe it was led by FDA investigators. They had a publication in Annals of Oncology where they discuss evaluating what data is being used, what external data is being used, and that being another piece of the puzzle, and making sure that the data that's being used as an external control arm is fit for the purpose. And they specifically speak to, you know, what the type of data source is, whether it's something like a clinical trial data set, where we at least think it's high quality data that was done in a curated and well-defined way in a defined population, or if it's from other sources. And I do think they emphasize the fact that it is context specific, and in the right scenario, if you can convince that this is a good way to generate evidence, that they will be open to it and will accept it as meaningful evidence for their purposes.
Mm-hmm. Mm-hmm. Frank or Jacob, do you have any comments? If not, I'll move to another question.
Well, I'll just briefly say,
go ahead, Jake.
Just reinforce this idea that you know, this is going to be a stepwise thing, and so what we're talking about here is fairly novel, although I think is something people have talked about, of course. When we're talking about single arm trials without a comparator arm, it's always really beneficial to then have some kind of comparator. But this is not to say that this would replace all randomized trials. And so what we're really talking about is where is the sweet spot of having a comparator data set that is more robust than historical controls. And as we're able to further establish within that setting, I think we'll really lay the groundwork for hopefully a broader work ahead that will lead the field faster and at less cost.
Thanks. Frank?
I was just going to comment on the nature of the data, but that may be one of your other questions, so I'll withhold.
All right, well, it's a good segue, Frank. I was going to ask is, control arms from clinical trials have certain, structure to it, and you know when the patient was randomized, so you have a time zero. I mean, it's a lot more structured than the so-called real world data, right? And there's lots of different kinds of real world data. So what's the, what are the specific challenges with the real world data, and what's the best practice? How do you manage the extra complexity of real world data? Because certainly real world data is often more available.
Right. So I've had extensive experience doing randomized trials in real world data, so-called pragmatic trials, and I've learned to appreciate some of the differences in the data sources. So if you real world data, I think people have to be very careful to realize that real world data, electronic health record data, claims data, this seems obvious, but sometimes you have to go back to first principles. It wasn't collected for the purpose of research. So it was collected for patient care or billing or some other reason. So if you're running a clinical trial, you have a clinical trial data set and you have the protocol. For instance, if there's a value that is blank, it's truly missing. You were expecting that value to be there. When you're looking at a real world data set, the concept of missing data is a bit ambiguous, because if it wasn't collected by the healthcare provider because it wasn't deemed necessary on that day or at that visit, it's not missing. It's what we call informed absence. In other words, there's a probability model you can actually calculate. One of my colleagues has done. So when you look at these data and you try to match them, you have to be very careful when you're matching patient characteristics at baseline. Probably okay, because you can extract those fairly well. But if you're looking at it from a longitudinal standpoint, the concept of missing data, data latency, how data are collected, how they're connected is a challenge. So my advice, if you're going to get into that business, is to spend some time understanding the provenance of real world data. How is it collected? Where did it come from? Claims data are usually pretty complete because the billing people want them to be complete, but they're not very deep. They're very shallow. Electronic health record data are very deep, but they're not necessarily connected in the way you might think. So it's just important to think about, people enter this with thinking real world data looks like the clinical trial data set, and it doesn't. It doesn't mean, and it also doesn't mean the real world data is of poor quality. It was of good quality. It could be of poor quality, but it was of good quality for the reason it was intended. So the fact that there are blanks or gaps, as I call them, in the data doesn't make it poor quality. It just means that the person collecting it didn't really know you were going to do research on it, so didn't bother collecting that information.
That's right. Jacob, you have any thoughts on this?
Yeah, this really seems like an area of tremendous potential. And I think for that reason, a lot of people are excited about the possibility of it. But there really are some real pitfalls in there, too, that need to be managed, or some challenges, I'll say. One, Frank just outlined a big one. And this is something we're really looking to get some further data on as well, looking at real world data compared to clinical trial data and seeing the differences that may exist. One big challenge to that is what Frank just outlined in the data that's available, and the one that I think is most kind of the biggest one really is functional status, where when we're enrolling patients in clinical trials, they have a defined functional status. Now, there is plenty of data about that functional status not being as hard a measure, and that there can be some variability in what functional status the clinicians are providing, but it is still a provided field within clinical trials. There are certain lab values that are going to matter for some diagnoses compared to others. And so, the challenge is, how do we get a data set that is as complete enough that we can really feel comfortable about our having these matched controls, as opposed to just general data sets. Now, these are all challenges, and it may be that there are specific groups that will need to collect some of this data going forward to be able to better prepare for analyses like this. But right now, what we're doing currently actually is kind of historical controls, or more general kind of comparisons. When there's a single arm study that goes for FDA approval, well, even if there's not a formalized comparator arm or a historical group, there is kind of the general knowledge of the field itself. And so I think the challenge is not so much that we have the perfect system right now, but how do we take steps forward in a way that is more trustable, with comparators that are more relevant than these kind of broad historical perceptions? Because that's really what serves the basis right now in many cases, is a historical perception, not even a historical control. And then when we're bringing historical controls in, this is broad data sets where we're not even taking the personal data points that we're describing or that we're hoping to do. And so there are absolutely some challenges to then setting a more robust comparator that's a more objective way of trustably comparing our experimental arm. And so these are steps to really keep in mind as we're building that framework forward. But there's tremendous opportunity there if we can really better define how to do that.
Thank you, Jacob. I see a hand up. I think this is a real one. Frank?
Yeah, it was a real one. So I think just one other follow-up comment. I think this issue of trying to link to real world data is particularly an issue in the safety arena. I think you can, for a lot of the reasons that Jacob pointed out, you can probably infer efficacy by looking at some things like survival and other things. But remember that in a clinical trial, adverse events are largely solicited. And so you get a very different array of adverse events. When you look in a real world data set, nuisance adverse events may not be recorded. The patient's thinking about other things. Nobody specifically asks if you have any adverse events. It might say if anything bad happened, but that's a different level. So you do have to think about, when you're trying to compare safety from where you're running a trial, where you're actually prospectively collecting information, and then trying to match that to information that's in a real world data source, has a number of sort of additional pitfalls.
Yeah. Thanks, Frank. What about registries? And what I mean by, in this context, a registry would mean real world data, maybe from an EHR or whatever, but then you go back and enhance that data by pulling all the records you can find on that patient, so that you do get all the prognostic variables, et cetera, et cetera. And then
in fact,
yeah.
I'm sorry, I interrupted you. Yes, that is an excellent sort of middle ground. It actually was even referred to in Pocock's original paper in 1976. And the idea that the best way to do these controls is to actually have prospectively collected data in that site. So it'll make the matching a lot easier. And you're right, a registry is it's less like real world data and more like a trial in a way, because you're controlling what gets collected. Doesn't mean you're going to find it all, but you're defining what gets collected when someone's in a registry, and you have some additional control depending on your informed consent infrastructure, but you have more control over what goes in there. So the concept of, you could modify the concept of what is or isn't missing, for instance, because you specifically went and looked for a variable and nobody responded, or they couldn't find it. So I think that is a great middle ground. In fact, randomized registry trials are very common. May not be appropriate in glioblastoma for the reasons Jacob said, but that's actually something that the FDA I know has been exploring. If you have registries, then you have a much more concurrent source of information.
So
That is
Lorenzo,
something that's come up in terms of, I would say, maybe, we think of pros and cons of whatever the data source is, whether it's real world evidence or trial data sets. I think for trial data sets, we keep talking it up, and I think rightfully so. I think the downsides that can come up, number one is probably for GBM, where it has the standard of care hasn't changed, is temporality, and then this idea that there may be changes in radiation technique, surgical technique, and supportive care that actually are changing outcomes over time, even though the treatment itself of chemoradiation is the same. And can we really use a failed randomized trial from 2006, 2007, and compare it to patients now? I mean, I think most people would be hesitant, are a bit hesitant of that. So, having one advantage of real world data, or at least prospective registries, as has been mentioned, would be that you at least have the temporality there that you wouldn't otherwise. In terms of trial data sets, of course, access and number of patients are other limitations in thinking that that real world data allows you to capture the information of many more patients and use that for your decisions.
Well, Lorenzo, I think in the GBM context, I think you have used both control arms of clinical trials and real world data. Is that correct, or has it been all real world data?
Yes, we approached both possibilities, and we were lucky enough that we had individual patient data for a couple of large randomized clinical trials. So that those were ideal data sets in order to understand if inclusion of external information is relevant. And then we compared with analysis that were instead based on real world data coming from Dana-Farber and also from other cancer centers. And actually there were differences. And when we were doing the analysis, we definitely realized that it was much easier to have errors incorporating into the modeling of real world data, so just because we had less experience and because there are key discrepancies that were previously described between clinical trial data and real world data. And one thing that I want to emphasize is that my impression is that we will extract a good amount of information, and it will be useful for clinical trial for real world data, definitely, but it is essential the opportunity to access and it should be easier to access clinical trial data sets. That is a key component of our possibility to understand if these architectures that leverage information from externally, from other data set, if it is applicable and if it is enough reliable. And it's also a key component of the communication with regulators. We have to prove that the approach is accurate enough, that there is not a large component of bias. So, and this effort, the access to randomized clinical trials that have completed and the possibility to do reanalysis of this large randomized studies, it is essential. And it is a key challenge, I think, of the overall enterprise of promoting the use of external information into the design of clinical trials.
Very good. Anyone else? What I'm thinking about now is, if you're making your submission to the FDA based on randomized trials, the very traditional way, and it's in an area where the doing clinical trials is relatively not problematic, then you can really specify the analysis or the experiment and the analysis plan and have a fairly unambiguous, this result means the drug works, this result means we didn't find activity. It seems like with the use of external controls, there's always going to be an element and a significant element of, well, that's all logical, but every case is different, every disease is different, every external control arm that you access is different. So we'll just have to wait and see what comes out. Is there a way to make this more systematic than where I think it lays right now?
Well, I think there's still a lot of work to do in kind of laying the groundwork on some of this, but I'll point out, there are different trials that are most important in different settings. And so when you're talking about a disease where there are plenty of individuals to enroll, the most robust, I think, that everybody would trust is a large randomized trial with clear outputs and all of this. When you're talking about something that's in development, a drug that is promising, and we want to get a sense of whether there's a signal for this and whether this is something that how the stats should be done, and we're talking about a phase 2, and in some settings by using an external control arm, we can enroll that more quickly. There are other settings where trying to enroll at all is extremely challenging because of the rarity, and we've outlined there where an external control arm provides some framework within which to analyze those results, which are not really possible otherwise. And so, one important point is that I don't think there's any one design that is going to be across the board the right call. But rather different settings where this is going to make far more sense than others.
Frank?
So you've actually put your finger on an issue that is of interest to the regulators. So in the clinical trials world, one of the major steps forward in transparency was the requirement to register trials back in 2005, 6, 7. And so any component of a trial that is called a trial or has some randomization will have a defined time frame in clinicaltrials.gov. So I can tell when the trial started, I can tell when the protocol was written, and I can therefore then trace when the analysis plan was created to answer your questions. One of the challenges for the regulators is when real world data are selected, and this also does contribute to the potential for type 1 error problem, I think, which Lorenzo said, and Kurt Veal has outlined in his publications. With all the best of intent, I can register an analysis plan, but even when I do that, the regulator is still going to have some level of suspicion. Well, how many times did you analyze the data before you wrote this analysis plan? Because I can't tell you when the data set was extracted. There's no legal or regulatory timestamp. And I think that's forever going to be a challenge. I know Jesse Berlin and others have tried to come up with registration issues for use real world data, observational data studies. But that is, I think, another piece of advice for people if they're going to interact with a regulator. The earlier you do this, the better, so you can have have more integrity to the process to answer some of the questions you're saying, because otherwise the regulator has every right to say, well, that's interesting, and I love to believe you, but how can you prove to me this is actually the first time you looked at these data? So.
And that, this raises a topic too. I guess I'd point out that one concern, I think, from a regulatory standpoint, is that that groups may know the benchmark that they have to beat to be able to have a positive study. And so on the speculative side, this is the skeptical side, I think this is one concern. And so one thing I'd point out is that with this external control arm that we're discussing, there is no static benchmark to beat. This really is going to adjust. I mean, the control arm will adjust depending upon the group you're enrolling. And so if you're enrolling a population of patients that are prognostically better, then your matched controls are therefore going to be prognostically better. And so your control arm is going to look better in that setting as well. And so this concept itself, there is no static control arm to this. It does adjust based upon the group you're enrolling as well.
Yeah. So great. We're going to bring this to a close and thank the panelists. A great job. And I hope all of the attendees also profited from listening to your expertise. I think one of it might have been you, Jacob, right at the beginning, who kind of framed this, right? So at one of the end of the spectrum, you do a single arm trial and you have sort of an ad hoc, here's what I think it would have been, or here's what I think the standard of care delivers. And then on the other extreme, you have the traditional large randomized clinical trial. And there's so much space in between, right? And most of the real important cases where we need a paradigm, a new paradigm, an improved paradigm, most of them are in between somewhere. They don't really fit either extreme. So I think it's a very rich area, and I really appreciate having you, making up this task force and already making significant progress. And I think we'll see a lot more as we go forward, because now these ideas are being implemented, and we will get results and we'll get reactions from regulators, et cetera. So it's still a really rich area of interest to many people. And with that, thank everybody, and have a great rest of your day. Cheers.
Thanks, Bill.
Thank you.
Thanks.